Hello everyone. Am I audible as well as visible? Let me know. Hello everyone. Hello everyone. Am I audible? Hello everyone. Am I audible? Okay. Let us start. Let us start. Hello everyone. Welcome to Unacademy Future Doctors. Here in this channel, we are posting videos for MBBS students. Okay, so anybody in your group, whether your senior, your junior, your family or friends, those who are appearing for first MBBS or other MBBS university exams, you can share these videos with all your friends or juniors. Okay. Now let me explain something about our subscription. Let me first introduce myself. I myself Dr. Dipti Kariya. I'm MD PhD Physiology, Associate Professor, and I'm educating MBBS, nursing, and physiotherapy students. I have more than 17 years of teaching experience. Now, let me explain about our subscription. We have two subscription, one that is plus subscription. Here in plus subscription, you can choose the best from the best. In plus subscription, you can access both live as well as recorded classes. You can study on the device of your choice. You can learn from India's top educators for your medical exams. You can access question bank with more than 25,000 questions. You can compete in the live test as well as quizzes, and within 12 months, they are coming with our printed notes. Okay. Next one, that is iconic subscription. Iconic subscription, in that you can access to the best from two of the best. Okay. So which are these two of the best? One that is Unacademy and Prep Ladder with Unacademy. Features I've already discussed. And second one that is Unacademy and Prep Ladder with Prep Ladder. Features are here you can have clinical and integrated essential video lectures from our dream team. You can have question bank 3 with active guidance, system tags, and much more. You can have rapid revision, snapshots, treasures, and 2021 dream notes. Okay? So you can subscribe from your mobile, either plus or iconic subscription. You can use my code DIPTI10 and you can get 10% discount on the subscription. Another thing, longer the duration of subscription, cheaper are the rates. Okay, so this is all about our subscription. And in this channel, we are posting all the videos which you can access freely. Okay, so if you are MBBS exam going students, that is all this video will be very helpful for you people because of COVID and all. Your studies will suffer. So this is a blessing for you all. So let us start our topic now. So today we will discuss in last class, we have discussed about humoral and cell-mediated immunity briefly, as well as we have also discussed about antigens. Now, today we'll discuss about antibodies. In your lower standards also, you must have learned about antibodies. Okay? Now, which are these antibodies? These antibodies, they are the proteins which are produced by plasma cells. Plasma cells are formed from B lymphocytes. So B lymphocytes, they form plasma cells, and plasma cells, they produce antibodies. Okay? And these antibodies are produced when antigen enters in our body. Okay? Now, these are gamma globulin. You all of you are knowing that albumin, alpha 1, alpha 2, beta, and gamma globulin. These are different plasma proteins from which gamma globulins they form antibodies. Okay? Now, structurally, now let us first discuss about structure of antibody. These are immunoglobulin and they have four polypeptide chains. You can see here. This is 1, 2, 3, and 4. These are four polypeptide chains. And they form 20% of total plasma proteins. Normal total plasma protein, if you can remember, normal value is 6.4 to 8.3 gram percentage, from which 20% are immunoglobulin. Okay. Now types are, I think all of you are knowing names: IgG, IgA, IgM, IgD, and IgE. Okay, so these are immunoglobulins. Okay, names of the immunoglobulin. Now today will you try to remember the whole topic in the class, so it becomes very easy for you at the time of exam to remember and to write down. Okay, I'll try to make it very easy. So now, starting with the structure. So structure, first of all, you can say that here, this is, as I told you, there are 4 polypeptide chains. One, this is second, third, and fourth. 2 heavy chains and 2 light chains. Okay. Molecular weight of this immunoglobulin or gamma globulin, that is from 1,50,000 to 9 lakhs. Okay. Shape is Y-shaped. You can see here, made up of 2 heavy chains and 2 light chains. This is heavy and this is the 2 light chains. So this one is heavy. This is light chain. Okay. Now heavy chain is having 400 amino acids, double than light chain. So amino acids here 400, light chain has got 200 amino acids. You can see the size is half. This is light chain, this is heavy chain. Okay. And they are held together by sulfur linkages, disulfide linkages. You can see here double sulfur, S2S are there. So disulfide linkage. And they have 2 regions, one that is constant region and second that is variable region. So variable is this one. This is variable. Why variable? Because amino acid sequence is different here. And constant because amino acid sequence is constant. Okay. Now, let us start with the heavy chains. You can see here, these are heavy chains. Molecular weight of heavy chain, that is about 50,000 Dalton. Okay. And structurally and autogenically, they are different for different immunoglobulin. As we all know, IgG, IgA, IgD, IgM, and IgE. They all have different heavy chains. Okay. So you can see here, easily remember, if it is IgA, alpha, A for A, D, delta, E, epsilon, G, gamma, and M, mu chain. Okay. And so you can say that heavy chains are identical for the particular immunoglobulin, particular antibody. Okay. So if your antibody is IgA, chain must be alpha chain. Okay. Now, heavy chains, there are 4 loops of 110 amino acid. You can see here. This is one loop, second, third, and fourth loop. So 4 loops of 110 amino acids. So total 440 amino acids are there. Okay. Then here you can see VH. This one is variable region. So it has one VH region, one VH, and 3 CH. CH1, CH2, CH3. Okay. So one VH and 3 CH region. Okay. Now, constant region. Why constant? As I told you, amino acids are same. Another name for the constant region, that is FC region. FC means fixation of complement. Here complement binds. What is complement? We will discuss in detail. Okay. So this portion binds with complement system. Okay. This one. Next is variable region. Variable region, why is it known as variable? Because different amino acids are there here. It is also known as FAB, fraction for antigen binding. Antigen binding fraction of antibody. Okay? This is antigen binding site. Okay? Now, light chain, if we are talking about this one, this is light chain. You can see here. These are 2 light chains. Molecular weight is half than heavy chain. Molecular weight of heavy chain is 50,000. Here, this is 25,000 Dalton. Okay. And there are only 2 types. One is kappa and second is lambda. And they are present in all the immunoglobulins, kappa and lambda chains. The ratio of kappa raised to lambda, that is 2 raised to 1. Kappa chains are always double than lambda chain. Okay. Here, same way as heavy chain, here also variable and constant region. You can see here, this is variable region for light chain and this is constant region for light chain. We have discussed here variable region for heavy chain, constant region for heavy chain, constant H1, H2, H3. So this way. So it has also constant and variable regions. Okay. In light chain, only 2 loops are there. In heavy chains, we have discussed 4 loops are there, each having 110 amino acids. Here, 2 loops, each having 110 amino acids. Okay. Now, we will discuss one by one immunoglobulin. IgG, we are starting with IgG. They are highest present. 71% of all the immunoglobulins are IgG. So the concentration is 0.8 to 1.6 gram per deciliter. Molecular weight is 1,60,000. It has heavy chain, that is gamma. As I told you, G for G, IgG gamma chain. Light chains are kappa and lambda. Okay? Now functions. These antibodies are antibodies of secondary response. You can see here. This is primary response, this one, and this is secondary response. Okay. So what is this primary and secondary response? Let us explain. Primary response is when an antigen enters and our body produces antibodies for the first time. The response produced, that is primary response. This one. Okay. So in primary response, amount of antibody production is less. You can see here only this many antibodies are produced. Whereas in secondary response, large amount of antibodies are produced. Because same antigen enters in our body, so our body produces large amount of antibodies. And this is the basis of vaccination. How? Because when we vaccinate an individual, we are producing primary response. Okay, suppose कोरोना कोविड की वैक्सीन ली है तो क्या होगा, ये एंटीबॉडी बन जाएंगे. ठीक है. फिर नेक्स्ट टाइम व्हेन एक्चुअली वायरस attack happens to our body, this large amount of antibodies are produced, and these are IgG type. Okay, and they are distributed equally between blood and tissue fluid. Another characteristic of them, that is they can cross the placenta. You can see these are IgG. They can easily cross the placenta, they can enter in the fetus. And this IgG antibodies, those who are crossing, entering the placenta and producing reaction, that we have discussed in hemolytic disease of newborn. Now, we discussed hemolytic disease of newborn. This of newborn, the antibodies are IgG type, they are Rh antibodies. Okay. Another action of this IgG, they act as opsonin. Now, what is this opsonin? Antibody or complement protein. You can see here, these are antibodies, this all. Okay. So, antibodies or complement protein, when it binds with antigen, it increases phagocytosis. So it makes phagocytosis easy. So that is opsonization. So also their function is for opsonization. Okay. So these are the functions of IgG. Next antibodies are IgA. Less than IgG. IgG are 71% total. Okay. IgA are 22%. Okay. Value, that is normal amount, is 140 to 420 milligram per deciliter. Molecular weight is 1,70,000. Okay. Here they have heavy chain, alpha chain. This one heavy one is alpha, A for A, and light chains are kappa and lambda chain. Okay. Now these are known as secretory antibodies. Secretory means they are present in the secretions. Secretory antibodies. So secretions like here saliva, intestinal secretion, respiratory secretion, breast milk, they all contain this IgA and they provide immunity. Now here other characteristic is they cannot cross placenta. Okay? So they do not provide immunity to the baby before birth. After birth, because they are present in the breast milk, they provide immunity. Okay? Another thing, they lyse the bacteria in presence of the enzyme lysozyme, which is present in the secretion. So they destroy the bacteria that way. Next is IgM. They are the largest one antibody. They are known as pentamer. 5 units are there. 1, 2, 3, 4, 5. All the 5 are attached by joining chain, J chain. Okay? 7% only, they are 7%. Concentration is 50 to 200 milligram per deciliter. Molecular weight is 1 lakh for each chain. Here, heavy chain is mu chain and light chains are kappa and lambda chains. Okay. Function is they are antibodies produced in primary immune response. You can see here, as we have discussed, when first time antigen enters in our body, antibodies produced, they are IgM type. This one, this. Okay. Another function of them, that is, sorry, you can see here. Sorry. Another function of IgM, that is they activate complement system. Okay. You can see here, this is IgM. When your antigen binds with this IgM, this is IgM, this is antigen. Antigen binds with antibody IgM, and then only complement, this is complement protein, they are activated. So they play an important role in the activation of complement system. Okay, and they are more effective than IgG antibody for lysis of the cell or lysis of the foreign cell. Now, next is IgE. They are present in very small amount. Okay, you can see only 0.03 milligram per deciliter. Their molecular weight, if you can remember, that is fine, 1,85,000. E for E, epsilon heavy chain is there. And light chains are kappa and lambda chains. Okay, you can see here, this is the structure of IgE. Okay. These are heat labile and skin sensitizing antibodies. These are responsible for hypersensitivity reaction or allergic reaction, you can say. Hypersensitivity or allergic reaction. There. You can see here. This antibodies, IgE, they are present on mast cells and basophils. And when your antigen or particularly allergen, when this allergen binds with IgE, then this mast cells they rupture and histamine, serotonin and other substances are released which are responsible for allergic reaction. Okay. Next is IgD, immunoglobulin D. These are also present in very small amount, 3 milligram per deciliter only. Molecular weight 1,50,000. Heavy chain D for D, delta chains are there. Light chains are kappa and lambda chain. Okay. Now, this function of them, that is, you can see here, they are present on the B lymphocyte. B lymphocytes, they have IgM and IgD. So, what happens here? They are important for recognition of antigen. Here, antigen binds with this IgD and it is recognized. Then B lymphocytes, they produce plasma cells and from which other antibodies are produced like IgG or IgM. So for this production, we require this IgD. Okay. Now, development of lymphocytes. All of you are knowing that all the WBCs, they are produced from, all the RBCs, WBCs and platelets, they all are produced from pluripotent hemopoietic stem cells. Okay. So here, from this, you can see here, from this pluripotent hemopoietic stem cells, we have discussed, there are lymphoid stem cells are produced. And from lymphoid stem cells, lymphoblast, and from lymphoblast, lymphocytes are produced. Now, this are, you can see here, so they are produced in the bone marrow. Okay? Now, from this bone marrow, T lymphocytes and B lymphocytes now they are processed at different places. So the cells, those who are designed to develop cellular immunity, or you can say T lymphocytes, now they they arise from bone marrow and then they pass to the thymus. And in thymus, they get preprocessed. Okay? And then they will go to circulation and lymph node and all. Whereas this B lymphocytes, they are processed when they are in fetal life, intrauterine life. These B lymphocytes are processed in fetal liver, sorry, in fetal liver and bone marrow. And after birth, they are processed in bone marrow. And after processing, they pass to the circulation. Okay? Now T lymphocytes. T lymphocytes are processed as we have discussed in thymus. This is the thymus. It is structure present in the neck. And where do they pre-process? The time of processing, that is just before birth and few months after birth. Now, what is the importance of processing? Here you can see this T lymphocytes. They are made. They come to know that this is foreign protein and this is self protein. So these T lymphocytes are not being produced against self protein. So T lymphocytes, they learn that this is our body protein and this is foreign protein. Bacteria are also made up of protein. Our body is also made up of protein. This is just an example. Suppose there is newborn baby. He or she is not knowing anyone, but within few months, he or she starts knowing that this is father, mother, this is unknown. The same way, T lymphocytes also starts knowing that this is our protein and this are foreign protein. So this is known as processing. There is one hormone thymosin. This hormone thymosin is secreted by thymus gland and it accelerates proliferation. Proliferation means multiplication of T lymphocytes and activation of T lymphocytes. Okay, and they also increase the activity of T lymphocytes. Now, you can see here there is pre-processing of T lymphocyte, and because of pre-processing, they develop extreme diversity. Diversity means differentiated in different varieties of T lymphocytes. Okay? And one thymic T lymphocyte develop reactivity against only one antigen. 1 to 1. Okay, one type of lymphocyte produces T lymphocytes from particular antigen. Okay? Now, so you can see here, thymus makes certain that T lymphocyte living will not react against self antigen. You can see, suppose our body produces T lymphocytes against self antigen, then they are destroyed, apoptosis, destruction. Okay? And they are destroyed by our process that is known as pre-processing, or that is known as self-immunological tolerance. Okay? Now, types of T lymphocytes. Okay, there are mainly 4 types of T lymphocytes you must have learned in your lower standards. Helper T cells, also known as inducer T cells. Other names are CD4 cells or MHC2. Second, cytotoxic T cells, also known as killer T cells. You can see here, these are killer T cells. These are helper T cells. Okay? Or CD8, because they have this CD8 plus proteins, okay, on the surface, or MHC1. Third, that is suppressor T cells, or you can say regulatory T cells. These are, and memory T cells. So first they are processed in the thymus and afterwards they are stored in the lymphoid tissue. Okay? Lymphoid tissues are the site where T lymphocytes are stored. Okay. Now, so you can see here, this is again the same thing. Fetal life lymphocytes are produced from bone marrow. Some of them T lymphocytes, those who are producing T lymphocytes, they enter in the thymus and they are differentiated in 4 types: memory, cytotoxic, suppressor, and helper T. Whereas some enter in the liver and spleen and also remain in the bone marrow, and they are differentiated into plasma cells and memory cells. They produce antibodies. Now, next is B lymphocytes. B lymphocytes are discovered first in Bursa fabricius, that is present in the birds, and therefore they are named B lymphocytes. But this Bursa is not present in human beings. So this lymphocytes, they are processed in bone marrow in human being, not in the bursa. Okay? And after processing this B lymphocytes, they migrate to lymph nodes and they are stored there. Now, from this B lymphocytes, there are production of plasma cells and B cells, memory B cells. This plasma B cells, they produce antibodies. Okay? This plasma B cells, they are, that is, you can say, here you can see B lymphocytes. When antigen enters, this B lymphocyte, it becomes large and having large amount of rough endoplasmic reticulum, and they form plasma cells. Now, these plasma cells, they produce large amount of antibodies. Okay? And in lymph nodes, some lymphocytes, they form memory cells. Now, this memory cells, memory B cells, they remain in this lymph nodes for a long period of time. Okay? They remain in the lymph nodes for a long period of time, and they produce lymphocytes and they produce antibodies on second and subsequent exposure by the same antigen. Okay? So this is about here different B lymphocytes, and they are stored, as we have discussed, they are stored in the lymphoid tissues. Which lymphoid tissues? Lymph nodes, spleen, bone marrow, and GIT. Okay? All they have storage of this lymphocytes. Now, this B lymphocytes, how they differ from T lymphocytes? Now, this B lymphocytes, they secrete antibodies. You can see here. And T lymphocytes, they produce T lymphocytes. Another thing is B lymphocytes, they have greater diversity. Okay? Diversity means they are differentiated more as compared to T lymphocytes. Multiple antibodies are produced from one plasma cell. Okay? So this is the difference. So this is all about our antibodies. Now, let us write down some questions from the topic. Okay. Till date, what we have discussed, some of the questions may be asked in your university. Write down all these questions. You can write down from the slide. So here, first, that is describe the physiological defense mechanism. First question, describe the physiological defense mechanism. Okay, in that you can write down about immunity, types of immunity, innate immunity, acquired and innate, which are the innate immunities, body surfaces, secretions. Okay, and in acquired also you can write down about active and passive, natural and artificial, active and passive immunity. Okay, now next question, that is innate immunity or natural immunity. Okay, so in that you can write down different natural immunity. We have discussed body surfaces. We have discussed in previous video. You can go through that video of immunity. Okay. Next is, what is immunity? As we have discussed, it is the ability of body to resist against pathological organism. Different types of immunity. Okay, that we have discussed. These are the types. You can write here. Here, same questions are written repeatedly. Why? Because it may be asked in this way. So you must know how to write down the answer. Okay? Then describe the role of lymphocytes in immunity. So you can write down here T lymphocytes and B lymphocytes and their role. Okay? Then discuss the immune mechanism in human body. In that you can write down all this immune mechanism. So these are long questions may be asked from the topic. Short questions are immunoglobulins. The whole immunoglobulin means you have to write down about all the immunoglobulins, IgG, A, M, D, E, their characteristic features, their functions. Okay, all that is written we have already discussed in the video. Okay, so you can write down from the video. If you are making running notes, साथ साथ में नोट्स बना लेते हो तो भी ये पूरा क्वेश्चन आप लिख लेंगे, बिकॉज मैंने पूरा ही कवर कर लिया है. तो ये लेक्चर के जो एंड में क्वेश्चंस आते हैं, दिस आर वेरी इंपॉर्टेंट. तो ये क्वेश्चंस आपको नोट डाउन करने हैं. स्ट्रक्चर एंड फंक्शन ऑफ आईजीजी, ये भी पूरा लेक्चर में डिस्क्राइब किया है, तो उसमें से भी आप वो लिख सकते हैं. आईजीजी का पूरा स्ट्रक्चर, the function, normal concentration, okay, then all that is given in the lecture. Okay? So this is all about today's topic. Thank you. If you like my video, please like the button. Please press the like button. You can share my video. You can subscribe the channel and hit the bell icon.